Hello everybody, here's Andreas from Azure Biosystems. Today I would like to talk about our brand new Azure Sapphire Biomolecular Imager. So the Azure Sapphire is a scanner which can be used for different types of applications, like standard Western blots or gels, but the machine can do much more. For instance, you could put on the glass surface different types of arrays, like protein arrays or cell-based arrays, and the webinar today is exactly about this topic. So let me take you on a little journey about array imaging on the Azure Sapphire Biomolecular Imager. Okay guys, let's get started. At the beginning, few brief words on our company. So we are a manufacturer from US. We are designing, developing and manufacturing quite close to San Francisco and the Silicon Valley. And the company itself, it's still quite young. It has been founded in 2013, but you see already, we have already over 12,000 users globally. We have already now over 3,000 imaging systems around the world in the field, and also the amount of publications is growing exponentially. And with the Azure Sapphire, we want to give you as a user more modalities, more applications. We want to give you more resolution, more sensitivity, more value. And the application part is indeed very important for us. We want to give you a very flexible machine which can be used for standard applications as said like Western blots or gels, but we want to give you with this device the possibility to do much more. And how do we make this possible? Well, we put inside different types of lasers. So the machine has included RGB laser light sources. It has also included near-infrared lasers. We have in addition put inside in the machine a phosphor imaging module and a chemiluminescence module. And when we look at the laser light sources, you see that up to 4 lasers can be installed into the Sapphire. So we have a blue, green, red laser and also lasers which are used for near-infrared. And at the end, it depends from which kind of field you are coming. If you are doing research more with RGB applications, then maybe a RGB only version is interesting for you. Or when you are coming from the Western blot field that we are doing just near-infrared Western blot, then maybe a Sapphire with these 2 lasers, near-infrared lasers, is interesting for you for the 700 and 800 channel. As a Western blot user, we can give you also the possibility to use a 685 nanometer laser, which suits better for the near-infrared part. Yeah. Or you are interested in a full equipped version, so we can put all lasers inside that you don't need to choose anymore between the RGB field and the near-infrared field. We have both included into the scanner. The unique thing about the Sapphire is that we have 3 detectors in one machine. We have installed a photomultiplier tube, we have inside an avalanche photodiode, and we have also a true CCD camera with a true CCD chip for chemiluminescence inside. So we have on this technology also already a patent in US. So you will only get a scanner with the 3 detectors, so PMT, avalanche photodiode and CCD chip in one machine only from Azure. And of course, we are now also trying to get this patent worldwide. We are using the photomultiplier tube for blue and phosphor imaging, so blue fluorescence and phosphor imaging. And the reason is here because the photomultiplier tube is giving you a higher quantum efficiency in the shorter wavelengths. as you can see here. On the other hand, we are using the avalanche photodiode for red, green, and near-infrared fluorescence. And the reason is because the avalanche photodiode has a higher quantum efficiency it's a longer wavelength, as you can see here. So at the end, it makes perfectly sense to have a machine with the avalanche photodiode and a photomultiplier tube. So you have combined, you will have as a user the full range of high quantum efficiency. In addition, we put inside into the machine a true CCD camera with a true CCD chip, and this is for chemiluminescence. So chemiluminescence is a still important technology, is right now still a bit more sensitive than fluorescence. Yeah. So let's say you have a complex protein mixture, you want to look inside, you know is your biomarker inside. So indeed, sometimes it makes sense to use the chemi here to look at the lower detection limit. And therefore we put inside this camera, which is sitting below the glass surface. In chemi mode, we have also the possibility to detect your visible marker. So when you have on your chemiluminescence Western blot a visible marker, the scanner will take a color image and then we will paste together the chemi image and the marker image. So the chemi image is really pixel by pixel on the right position. That's a great feature. The field of view of our scanner is 25 by 25 cm, which is for the most type of application, fluorescence applications, more than enough. Also for chemi, we have 16 by 13 centimeters, and this is also enough for the most types of chemiluminescence Western blots. The very specific thing about the Sapphire is that the Sapphire has a super high resolution, up to 10 micron resolution. That's really high. And important to know is here, this is not interpolated, it's really true resolution of 10 micron, which will give you the ability even to see single cells like motor neurons. The Sapphire is also very fast. It's scanning 15 centimeters per second. And the special thing is here that we are scanning with up to 4 lasers simultaneously. And this, you imagine, gives you really a speed boost. You don't need to wait anymore until the first laser is finished and then the second is scanning. We can scan with 4 lasers simultaneously. If you want to scan sequentially, then we have also this option in our device. The Sapphire is coming to you with an external computer, and you as a user at the end can choose if you want to have a laptop or high performance desktop computer with a great big 20-inch screen. Of course, included is also the capture software. Let's have a brief look how it looks like. So the capture software is really easy to use. Just 5 minutes in front of the machine and you know everything. So just put your specimen on the glass surface. You are selecting here the area. Then you choose your method of choice, the pixel size, so the resolution, the scan speed, and then you click scan. That's it. Very simple. So the machine will generate for you uncompressed TIFF files, and then afterwards you can go ahead with the analysis. Important to know is here that we are not forcing you as a user to use our analysis software. We have a very flexible format. As said, the machine is generating uncompressed TIFF files, which are not interpolated. This is pure raw data. And you as a user can choose what you want to do with this. If you like to use free software like ImageJ and use it so far and want to go ahead, no problem. you can take the tips and put it there and go ahead. If you want to have a professional analysis solution, then we can offer you our Azure Spot software. Yeah? It's very detailed, it can do a lot. You see it has different modules, like this Western blot and gel module, which is automatically, it can detect automatically your bands, it will do normalization, quantification, it will print a report for you, and much more features. Then we have also something like we call analysis toolbox. This is a manual module where you draw the boxes manual around your bands. Then we have also a module for arrays, like protein arrays, 96 well plates, 6 well plates, and so on. And we have also a colony counting module. But again, the software is optional, so you can choose. Let's look at the configurations of the Sapphire. So the Sapphire is very flexible, and well, depending on your needs, we will generate a customized machine for you. So when you are coming more from the RGB world, you can choose the Sapphire RGB. When you are coming more from the near-infrared side and you are doing a lot of Western blots only, then you can choose the Sapphire near-infrared. If you want to do both, Sapphire RGB near-infrared would be the right machine for you. In addition, we have optional modules like the chemiluminescence module, to do the chemi. We have also a phosphor imaging module, if you want to do phosphor imaging, and also a Q module. And what is the Q module? Let me explain. So maybe you heard already about the Western blot 2.0 story. So what is Western blot 2.0? Well, it's about total protein normalization. There's a trend in the market. People are going away from normalization against the standard housekeepers like GAPTH and beta-actin. And the reason is there because, of course, GAPTH and beta-actin, this is just one protein, right? So we're relying on normalization on one protein, and it depends on the cell cycle. The protein expression of these housekeepers will always go a bit up and down, right? So it's more robust in case when you have a complex protein mixture to put the normalization on all shoulders. I give you an example. You have a complex protein mixture with 7,000 proteins, So why not to put the normalization on all shoulders? So we can do this, and we will support you on this new trend. So you can have the Sapphire near-infrared for your near-infrared Western blots, 700 and 800 channel, and then we can build in for you the Q module. And the Q module is our green laser. The green laser is far away from the near-infrared. It's more on the left side. So the near-infrared part, 700 and 800, is clearly separated from our green laser. So there's no crosstalk at all. And we can use this green laser for total protein normalization. We have also our own stains, for instance, our Azure Red. And this configuration of Sapphire is called Sapphire near-infrared Q. All right. Now I would like to talk about the amazing additional research possibilities which are possible with our scanner. And the topic today, I would like to set the focus on the array imaging. So you will see examples from the fields, from clients during several of our demos in the field. I've recorded several arrays, and I would like to give you a brief overview here, what is possible. So let's see. The first images which I would like to show you are protein arrays. So indeed there's interest in the fields on protein arrays, and I will give you an example. So this is a protein microarray which we put on the Azure glass surface. So the format is like a microscopic slide, and we have scanned this here with 10 micron resolution, so full resolution. Here you see the protein array, and in each square there are 225 protein spots. So in total, these are here 3,600 proteins spots on this protein microarray. And let me show you the results. So this is in 2 channels, and you can see very nice the signal of interest, the red one, and the difference in intensities of the protein spots. I also like to watch this into black and white, the channel of interest only in black and white, so you can see it even a bit better. And now is the question, is the resolution here high enough for quantification? Protein microarrays, of course, we want to look at the densitometric analysis. So let's zoom here on 2 of them, of these 2 squares. You see here. And, well, the resolution is more than enough. Yeah. So there are enough pixels per protein spot for quantification. And we have already several clients which are very satisfied, and they are quantifying their data with the Sapphire. Yeah. Also, our analysis software, it has a grid, so you can put a grid around this. The squares will, the protein spots will be detected automatically, and we will have a readout of the intensity of each protein spot. Yeah, and then we can do the analysis afterwards in the analysis software and also in Excel. We have a nice Excel export in our analysis software. Let me give you a second example of protein microarray. This is the second one. Same method here that has been used, only the format of the microarray is different. This is a different format. Look at this. Also see here, I think around 3,000 protein spots. Okay, this is in 2 channels. Let's have also a zoom in and in black and white as before. So first in black and white, and we can also zoom in. And also here you can see the resolution is more than enough for quantification. Yeah. So we can clearly see each protein spot and the different shades of gray you can see here, yeah, the different intensities. So quantification can be done really easy here. This is a third protein array that I would like to show you, and this has been recorded with 20 micron resolution, so even not the full resolution. But also here, this is already more than enough, yeah, for quantification. This is in black and white, 1 channel. I can show you also here in the inverted channel. This looks also good. Yeah, and this is the color channel, the single channel. And now I would like to talk about a second type of array imaging, which is very popular in the field. Well, cell-based assays in microplates. So at the end, it's like a Western blot. We are just doing this directly in your cells. Very popular in the field, and I think in the upcoming years, this will become more and more interesting, and more people will do this because it's very convenient to do. So this cell-based assay, we recorded this with 3 channels in a 96 well plate. And the resolution here we used for scanning is the highest resolution, the 10 micron. So let me show you this. 3 channels, you can see here this, and yeah, very high resolution. And you can imagine when you zoom in here, you will see also a lot of details. Next example, this is 96 well cell proliferation assay. And in this case, I will show you a zoom image out of this 96 well plate, and we scan it here with 10 microns. So look at this. So while I'm showing you this, I want to show you here really that the resolution is so high that we really can see the single cells or the very tiny cell colonies. So let's have a zoom at this part. And you can see here, right? These are the really tiny cells or little tiny cell colonies in the well, which you can see here already. So you see the resolution is really good for a scanner. This example here, this is a 384 well cell array. And in this case, also, I would like to show you a zoom out of this 384 well cell array. And we have scanned this here with 10 micron resolution. So look at this. This is in 2 channels. This is in 3 channels. And what you can see here, well, we used black well plate. So a black well plate we used here. Yeah, only the bottom was transparent. Yeah, so I really can recommend to use this black plates. Yeah, and so you can reduce light scattering. You can imagine that when the laser is hitting a transparent well plate, yeah, then it's like a fiber optic cable, and the light will be everywhere. Yeah, so it makes sense to use these black plates to reduce this effect and to get very nice results like here. And also here in our analysis software, we can put a grid around these squares, and then you will get a readout of the intensity of each channel at the end. And you can do the quantification. And here at the end, I would like to show you here a 6-well cell array. So this, of course, is a bigger format, but we scanned here also with 10 micron resolution, so full resolution. This is one channel. And here's the second channel. I think this was the channel of protein of interest, GFP. And now I would like to show you the overlay of both channels. So what you can see here nicely, the red signal, this is the autofluorescence of your cells. And overlaid here the green signal, this is the GFP signal, which you can see in the well number 6 and number 8. Okay, guys, so I hope I could give you here some appetite what you can do more with our Azure Sapphire Biomolecular Imager. If you're interested to learn more about our machine, I recommend you go to YouTube. We have a nice YouTube video. Just type into YouTube Azure Biosystems Sapphire, and you will get to see this video. Also, I can recommend when you visit our website to go under Learn Webinars to our webinar videos. So we have different additional webinars for you here where you can learn more about our products. If you have any questions, please don't hesitate to contact us. You can always write to info@azurebiosystems.com. So we will support you. And so I would like to thank you so much that you attended this webinar. It would be a pleasure to meet you in your own lab to show your own our Sapphire with your own samples. So thank you for attending once again. Have a nice day. Take care and bye-bye.